All right, hello folks. We're going to talk about blood specimen handling. This is chapter 11. Along with handling of specimens, we're going to talk about special testing procedures, transport of our specimens, and also some reasons why our specimens may be rejected or need to be recollected. So let's talk about specimen transport first. Now this might be within our facility, or we could be talking about outside of the facility, whether we're collecting from different clinics offsite and bringing them back to the main hospital, or talking about mailing a specimen to a reference lab elsewhere. So when we're talking about transport within the facility, depending on the size of the facility and the equipment they have, this might mean us just walking the specimen to the lab. But there may also be other methods of transporting our specimens. Like the picture here, we have the pneumatic tube system. So with each method of transporting our specimens, there are pros and cons, and we'll talk about that also when we talk about transporting outside of the facility. So with a pneumatic tube system, for example, it's really great, it's efficient, it's right there, it's easily accessible. But if you think about a pneumatic tube system, it's using a vacuum to take the specimen from one floor to the next, for example. So we do also have to worry about how this affects our specimens. Too much agitation can cause things like hemolysis or other adverse effects that are going to alter our test results. So before any transport system is implemented into a facility, the laboratory is responsible for making sure that these tube systems, or I said any system, whatever it might be, what are the pros and cons? What are the adverse effects that these are having on our specimens? How can we minimize, prevent them? Or are there certain tests that we just know are going to need to be walked directly to the lab versus using, for example, this pneumatic tube system? So then we're talking about transporting outside of facilities. This is typically done by courier, which is a person who's going to go to the various sites, pick up those specimens and drive them back to the lab, or the mail. So when we're talking about a courier, there's pros and cons to this as well, and things that we need to be aware of. This courier, of course, has to be aware of temperature, timing, handling, and our job as phlebotomists is that we have to make sure that our specimens are packaged appropriately, obviously labeled appropriately too, but packaged appropriately to prevent for leaks and spills and breakages. Some packaging may require ice or dry ice or a cold pack to go with it. So these are all important and critical steps and roles that we as phlebotomists have to play in this as well. So when we're talking about mailing specimens, this also requires proper packaging on the part of the phlebotomist and preparation. Most times we're packaging and sending a lab or a specimen to a different lab, whether it be via courier or mail. We're going to be packaging our specimens in a plastic bag of some sort. Typically when we're doing this, we are going to have our labeled specimen put into a bag with some sort of absorbent material or absorbent sheet. This is there for if we have that leak or break or whatever, it's going to absorb that material. Typically, these bags have a separate pocket from where the specimen is contained for paperwork. And that is also to prevent that paperwork from being damaged, again, if there's some sort of break or spill. Our packaging, of course, has got to be sturdy. And just like with the courier, if we're going to send it in the mail, we as phlebotomists have to know if it needs to be packaged with a cold pack or dry ice, for example. It's really important that we document all steps in the packaging and transport process, because if there is an issue with the lab or specimen when it arrives, it needs to be able to be tracked back so that we can look at errors along the way and figure out how can we prevent any errors from happening again, or where did this error take place. Some specimens also need to be tracked for timing purposes, right? So, and that might be timing of the lab to make sure that it hasn't gone too long between when the lab was collected, when it was tested. And then also this is a very handy tool for providers and physicians to be able to track specimens, when they can expect results. All right, so I'm going to skip over this slide because we're going to talk more about timing, especially when we talk about the centrifuging specimens in a minute here. But basically timing, which we've talked about before, is important when we're talking about the transport of labs, whether it be an as soon as possible, a routine, or a stat lab. Again, as a reminder, stats typically we are taking immediately to the lab, and the results are expected within 1 hour. So we're going to talk about special handling of specimens. In particular, we're going to talk about when specimens need to be kept warm, need to be chilled, or when they're light sensitive. So certain specimens need to be kept warm. And this might actually start with the pre-warming of the tubes. And this is done by either keeping these tubes in a warm water bath or by wrapping a heel warmer around the tubes until we're ready to collect the specimen. So oftentimes, tubes that need to be kept warm are going to be collected in red top tubes that do not have any additives in them at all, or in our lavender top tubes with the EDTA additive. These tubes, after we collect them, remember they're pre-warmed, we're going to collect our specimen and we're going to deliver it immediately to the lab, in which point we as the phlebotomist are directly putting them into a warm bath or an incubator in the laboratory so that they stay warm and ready to be tested. There's also specimens, like I mentioned, that require chilling. These also can be pre-chilled. And then these specimens, once we collect them, we're going to put them immediately into what we call an ice slurry. So typically what this is putting the specimen into a bag and sealing it, and then putting it into the ice slurry. Now, why do we do that? Main reason is because we don't want that label from the moisture to get smeared or ruined, or then we're going to have to recollect that specimen anyway. So put your specimen in a sealed bag and then put it into an ice slurry, which is a combination of ice and water. With the ice slurries, we don't want to have it so cold that we're freezing our specimen, but we also need to have enough water in that mixture so that our tube can be evenly chilled. So let's talk about light sensitive specimens. So some specimens, depending on what analyte we're trying to isolate and test, like bilirubin, for example, is a good example of that, or carotene, light will break those analytes down quickly. So we have to protect them from light. This can be done pretty simply. Either we wrap the specimen in foil as soon as we collect it, or these micro collection tubes, like the one in this image, come in actually like an amber colored plastic, and those tubes themselves will protect specimens from light breakdown. There are some special handling considerations when we're talking about blood alcohol, forensic testing, and toxicology specimens. And these primarily have to do with the fact that these specimens can be used in court. So all of these specimens here are, well, I shouldn't say all of them, but especially the blood alcohol forensic, any one of them that does end up being used in court is going to need a chain of custody. So what a chain of custody does is it basically ensures the validity of the specimen from start to finish. It guarantees the validity, integrity of the specimen from start to finish. And that's going to start with when we, as the phlebotomist, are collecting our lab. We have to ensure that our specimens have not been tampered with in any way. If the phlebotomist does not follow the steps in the chain of custody, then we can't prove that chain of custody was withheld, and those specimens cannot be used in a court of law. So the chain of custody, again, has to be completed appropriately and completely and fully, or it's not going to work. There are usually typically several copies of that chain of custody. One's going to definitely going to accompany the specimen itself. One's going to stay in a patient chart, and the other one might be used in a variety of ways. It could be attached to the packaging. That would probably be the most common thing that you would have if you had a third copy there. Another important thing to keep in mind is that when you have collected a specimen that's going to have a chain of custody and is going to be used in court for some legal purpose, we need to make sure that those specimens are in a locked container and sealed so that they, again, know that nothing has been tampered with. Blood alcohol testing. This also involves a chain of custody and must have consent. So it's important to keep in mind here that somebody can refuse blood alcohol testing. And if the phlebotomist is to go ahead and proceed with blood alcohol testing, that can be a legal case against the phlebotomist for battery or assault for performing a test that was not consented to. And in addition, then again, you do a test that requires consent and you don't get it, it's not going to hold up in court. Forensic testing and toxicology testing, we're going to talk about that real quick as well. Forensic testing, usually when we think of forensic testing, that's trying to link one person to another person or a person to an object or an event. So this is typically done in response to trying to figure out a criminal proceeding, right? Something happened and we're trying to figure out evidence for it. So this is going to involve chain of custody procedures as well. So your work as the phlebotomist is, again, to make sure that we're following that chain of custody, filling out all paperwork appropriately. Nothing is being tampered with. And then when we're talking about forensic testing, we have to be even extra cautious as the phlebotomist to not let any contamination happen. So in these particular testing scenarios, we're making sure to wear gloves at all steps of the process to avoid that contamination. Toxicology specimens. When we're talking about this, we're typically talking about the study of poisons, drugs, or medications in the blood. An example that you've already learned about here is actually just the peak and trough levels of a drug to see those levels of the medication in the bloodstream, right? But we could also be testing for things like lead or mercury or other types of poisons in the blood system and what their levels are. So you want to make sure that you're being careful with the handling guidelines for toxicology specimens, again, in contamination. And if there's any chain of custody involved, making sure we're doing that completely and accurately as well. So we're going to talk about some different special handling considerations during venipuncture. And a couple of examples, we're going to use lactic acid specimens and coagulation specimens. So let's talk a little bit about lactic acid blood collection, because that does require some specific handling procedures when we're doing venipuncture. Some facilities actually require that you don't even use a tourniquet at all when you're collecting lactic acid. And that's because lactic acid levels can increase for really a few different reasons. One of them is that we've left that tourniquet on too long and too tight. Another reason is that vigorous hand pumping. So remember, when we're collecting a venipuncture, we can ask our patient to squeeze and hold that fist, but pumping is actually going to falsely increase the lactic acid levels as well as alter some other analytes. And then exercise, we've also mentioned that, especially in that outpatient setting, you have somebody who, for whatever reason, decided to run a marathon and then come in for their labs. You want to have them sit for about 20 minutes before drawing their blood, because those activities can falsely change different analyte levels in the blood, and in lactic acid case can raise it and give false levels. Like I mentioned, the tourniquet, if you guys remember, we don't ever want to leave a tourniquet on for longer than a minute. Typically, when we're collecting lactic acid, we're going to use the gray stopper because it's got that sodium fluoride additive in there. And we're going to put it in a mixture of ice and water, so that ice slurry mixture, and transport it right to the lab. Lactic acid specimens really need to be run as soon as possible so that we get the most accurate results. And you might be asking yourself, why do we use the gray topper with that sodium fluoride additive specifically? Well, if you recall, that sodium fluoride additive helps prevent the breakdown of glycolysis. So what does that have to do with lactic acid? When glucose breaks down glycolysis, it turns into lactic acid. So if that glucose has been allowed to break down too quickly in our specimens, it's going to give that false level of elevated lactic acid that's not as true to the real picture in that patient when we first drew it, right? In addition, the ice bath that we put that tube in, the ice bath also helps to, along with the sodium fluoride, the ice bath also helps to slow down that process of glycolysis. All right, so let's talk about coagulation studies for a second here. So coagulation studies, we have to be careful about timing with these as well. Coagulation studies also require a couple of special handling procedures. We want to make sure we're following the order of draw, because remember, if we get any contamination from additives, that's going to cause our coagulation studies to be off. Remember, the light blue tube top for coagulation studies is always drawn before any tubes with different additives. Along with following the order of draw, when we're collecting for coagulation studies, if you recall, we talked about in phlebotomy, we tend to use needles with the gauge of 21 to 23. When we're doing coagulation studies specifically, we'd want to use a needle with a larger gauge. So really want to use a minimum of a 21 gauge bore needle for coagulation studies. And we want to minimize that time that tourniquet's on as well as much as possible. Definitely never longer than a minute, but the more we can minimize that tourniquet time when we're doing coagulation studies, the more accurate our test results are going to be. And if you recall, when we're doing coagulation studies, those tubes definitely need to be inverted in order for that mixture to be mixed with that, or the specimen to be mixed with the additive appropriately. All right. Now let's talk a little bit about separated specimens. So this is when we start talking about centrifuging our specimens, which we've touched a little bit on here and there, but not really ever talked about what is it and what is it doing. So a centrifuge is, the spinning. Basically, it is the spinning down of specimens to separate the cells in the blood from the liquid portion of the blood. So you can use either the serum or the plasma tubes. But if you remember, serum is obtained from tubes that do not contain an anticoagulant, and that would be like our red or gold top tubes, whereas plasma is obtained from tubes that contain an anticoagulant, and that's like our light blue tube tops or our green tops with the heparin in them. When a lab specimen requires separation, this has to be done within 2 hours. If the blood cells are left too long in touch with that serum or that plasma, it can alter the results by altering these different levels of analytes, especially glucose and potassium. Those are really affected by sitting too long without being separated. All right, let me see if I've missed anything. So let's talk about this centrifuge a little bit more. We know that centrifuges use the spinning motion to help separate. They basically use gravity. So centrifuges have different speeds. They have a dial that will talk about speed and time. Most specimens, let me see if it's on here. There we go. Most specimens are centrifuged at 1,000 to 3,000 revolutions per minute, and typically for 15 minutes. Of course, you're always going to go by the manufacturer or your standard operating procedures to do this properly for each specific test, but that's a good general rule there. You can spin different size tubes at different times, but you have to make sure that that centrifuge is balanced. So what we mean by that is, if I have 2 tubes, okay, let's say I have 4 tubes. 2 of the tubes are filled right at the halfway mark of that tube, and the other tubes are filled all the way to the top. So the 2 tubes that match have to be on opposite ends of each other, just like in this image. So I can mix all 4 tubes. I can centrifuge all 4 tubes at the same time, but it has to be balanced. If it's not balanced, it can actually, be hazardous to us as the phlebotomist stand next to it. It can break, it can shatter, it can, it'll shake kind of roughly. So we have to balance our centrifuge. Let's see what else here. All right. Okay, aliquoting specimens. So this is also something you may be doing. This is the process, of transferring a portion of a specimen into another container. We have to pay attention to details when we're doing this. We have to make sure that all tubes are properly labeled, because listen, if I have my labeled specimen that is in the lab now, and a portion of that is being transferred to another tube for further testing, what do I need to do to that second tube before I can move the blood or whatever portion I'm moving? It needs to be properly labeled. Super important. So make sure you have labeled specimen containers accurately before doing any kind of aliquoting. When you open any kind of tube stopper, it is going to be really important to either use an absorbent pad to block any aerosols from coming out, or possibly open under a fume hood. So we're talking about aerosols here. It's producing aerosols when we do that, and that can be a hazard to you. So again, Make sure you're using proper PPE if you're ever allocating specimens, proper PPE, absorbent pads, decrease your risk of being exposed to aerosols that might cause harm to you. So let's talk a little bit about specimen rejection. So laboratory specimens basically are only as good as, or the results I should say, are only as good as the specimens that are collected and tested. So as phlebotomists, we have control over specimen collection. We have control over the handling and the various pre-examination variables, right? Pre-examination errors are basically any error that occurs before the specimen is analyzed, all right? You, as a phlebotomist, we need to know some different causes for specimen rejection so that we can help to minimize them from actually happening. And I've got a list here of some common causes. Hemolysis, that's kind of one of the biggest ones that we see. This is, of course, that breakdown of red blood cells. When red blood cells are broken down, so this is going to be an example here. When in that picture, when the red blood cells are broken down, they release, they get that reddish color in the plasma. So when we see that reddish color in that top part over there, you can kind of see that reddish color and that mixed in with that yellow, that can actually affect the ability to run tests because of that coloring in the tubes. When hemolysis is present, it doesn't make it easy for us to test things such as potassium and calcium, so our results aren't going to be accurate and we're not going to be able to use them. Common causes of hemolysis, that would be you've cleaned the site, but you haven't allowed the alcohol to dry completely on its own. If we continue drawing, even despite when we see the presence of a hematoma forming, if we've done a capillary or dermal puncture and we squeeze too forcefully, that can cause hemolysis. If when we're inverting, we're not doing it gently and we're instead shaking or doing it too vigorously. If we're using a syringe method for transferring blood and we use force on that plunger, that can cause hemolysis. Or just roughly transporting our specimens from the collection site to the laboratory. Let me see what I have. All right, yeah. So another specimen that might get rejected is a clotted anticoagulated specimen. So a specimen that was not supposed to clot, and it did. If a test requires whole blood, plasma, or cells, it is collected in a tube containing this anticoagulant to prevent that from happening. But common causes when a specimen might clot when it shouldn't is we did the incorrect area of draw, right? So we got some additive in there that wasn't supposed to be in there. Failure to mix the tube, so we didn't invert our tubes like we were supposed to. Or if we're using the micro collection tubes, if you remember when we want those to not clot, we do after each drop, we gently tap it on a hard surface. If we use a syringe method and we didn't transfer our blood and syringe to the evacuated tubes quickly enough, that can cause clotting. Or another one, expired tubes. If our tubes were expired and we used them, they might not do what they're supposed to, and we might have clotting in our anticoagulated tubes. Some other specimen rejections is an incomplete collection, or you might see it written as quantity not sufficient, QNS. They basically don't have enough blood to do the test that they need to do. Sometimes you'll see quantity not sufficient when there will be enough blood in the tube, but the additive to blood ratio is off, so we can't use that blood or we'd get erroneous results. The citrate tubes, which is our light blue tubes, always have to be filled to the fill mark. They have to be filled 100% or we can't use them. Some other reasons for quantity insufficient: loss of vacuum during our venipuncture. Let's see, so, or maybe the tube didn't have the, well, most likely it'd be they lost the full vacuum. So maybe it was an expired tube and it didn't have that vacuum that it should. Let's see, what else? If you didn't remove the air from the butterfly tubing, and so you got some some air in the tube that wasn't supposed to be in there. the use of the expired tubes, not as the phlebotomist, not ensuring they get to the fill level. If the veins collapse during venipuncture and you just can't get any more out. If we're talking about doing a dermal or a capillary puncture, sometimes that site becomes clotted before enough of the specimen is even obtained. Remember how quickly capillary punctures start to clot. Some other things that might happen that might cause our specimens to become rejected. If a specimen is collected using the wrong tube, that's an automatic rejection. Causes of incorrect collections, procedure manual was not consulted. So if you don't know, if you're not sure, you've made a mistake, you didn't consult the standard operating procedures. Sometimes tubes get delivered to the wrong section of the laboratory for testing. They might get rejected there. Or sometimes, unfortunately, if the requisition itself was misinterpreted, and so we didn't do what we were supposed to do. The incorrect order of draw, of course. The correct order of draw is the most important thing we can do, especially when we're talking about coagulation testing. Sometimes a discard tube is drawn before that light blue tube, especially depending on the equipment that we're using for the draw. Let me see, hemoconcentration or contamination. Hemoconcentration and contamination can happen because of IV fluids running, right? So we forgot to turn off the IV fluids, or we drew from above the IV site. Let's see what else I have here. Let's see. So let's talk about icterus and leukemia. That's also in your book. I know I don't have it on the slide here, but icteric plasma and serum appear dark yellow or greenish yellow in color. And this is because of an increased amount of bilirubin. Bilirubin is made when red blood cells break down, or hemolysis increases your level of bilirubin. So if you see that kind of dark yellow or greenish yellow appearance in the test tube, this abnormal coloring there, just like when we have that red coloring in the test tubes because of excessive hemolysis, that can actually interfere with certain tests and allow them to not be run. Leukemia is the presence of abnormal amounts of fat in the blood. This can actually make the plasma or serum look cloudy and can interfere with certain tests as well, such as testing for hemoglobin levels. If special handling requirements are not followed, specimens are going to be rejected. If we don't have enough blood, specimens are going to be rejected. So just keep in mind that it is our job as the phlebotomist to make sure that we're doing these proper and special handling and transport procedures. All right, so there's do's and don'ts, right? Specimens can also be rejected because of inadequate or inappropriate or missing documentation, or for specimens not being labeled appropriately. So these are just some examples here. You can see the specimen tube under the do's. It needs to be neat, straight. You don't want it to be wrapped around and doubled over on itself. It can't be crinkly. I hope no one ever attempts that three tubes with one label. That's not going to work either. It can't be hanging off the bottom, and it can't be touching that stopper top, because that could be torn if it does need to be removed. All right, let's see what else. That might. Oh, it does talk about this. I'm sorry, you guys. I forgot I had this slide in here. But this is an example of that icterus specimen or the leukemia results. Let's see. And you can see the difference. And we talked about hemolysis earlier. So you can actually see the difference in the specimen that says jaundice there. That's that increased level of bilirubin. The leukemia specimen, that's an increased amount of fat in the blood. And the hemolysis, you can see the different colors. The range of hemolysis that has taken place. So the different varying colors of red there, okay? All right, so I think that's it. Yeah, that's it, you guys. So if you have any questions, let us know, and you guys are all set with chapter 11. Thank you.